And I must say, we must have had a very interesting session so far for the past few weeks. Today marks the 6th session of this cohort. I want to formally appreciate us for being part of the team from the first to the 6th session. It has been a wonderful experience having you all on board in this cohort. For the past 1 month, we have been discussing very, very interesting subtopics that will lead us to understand next generation sequencing. Today we have the last session, and we are looking forward to have our session wrapped up, and we move down to the next phase of this training. Mind you, this is the first cohort for 2021. The second cohort is going to come very soon. Today we are having our community manager to discuss with us a very germane topic for the last session, and also having hands-on. So Dr. Aditayo will be the one anchoring the session, and is expected to give us feedback on how far we have done so far. So, ladies and gentlemen, before we move down to discuss with Dr. Aditayo, I want to ask us to unmute our mic and let's have response so far. Let's see who wants to. If you have a comment or so, can you please unmute your mic so we can get to hear you? Yes, who do we have? I shouldn't be calling names. Nancy, who is first to join the live session today. Promise, Taiwo, Oboli, Dami, Banke, Adejumo, Ade, Abdulaziz. So, yes, do we have 1 or 2 comments so far with our experience with what we have on ground?
Hello, Mr. Sonny.
Yes.
Hi. Since Dr. Oboli is the class rep of this cohort, I think it behoves him to open the floor for feedback.
So, Dr. Oboli, Dr. Adita has chosen you, and we are good to go. Can you unmute your mic? Let us hear from you. What has been experience been so far? You're chatting it.
Is the one Dr. Nancy and others give us the. So, Dr. Oboli, I hope you.
Are you expecting to voice this? Dr. Oboli is saying I would say that this training has been an eye-opener for me. It has broken down concepts into well-sized bits that have really been helpful to me. Okay. Sorry, I'm in a noisy place. Okay, yeah, good one. Nice, good one. Apart from Dr. Oboli, do you have any other person that can give us comments on how the session has been so far? We want to hear from you so that we can know what we need to do better than what we are doing now. So,
hello.
Yes, I can hear you. Who are we talking? Can you speak up, please?
Yeah. Okay, this is Nancy.
Nancy, beautiful. You have the floor, please.
Yeah. Okay, I just want to say it's been amazing being among this cohort for the training. I joined, I was among the cohort for bioinformatics for genomic medicine, though I didn't have much time to follow through the training, but I followed through this one because of just a sneak peek I got from that one, and it's been amazing. And like Mr. Poli said, it's actually an eye opener, and it's kind of pricking my interest more in the field of bioinformatics. Actually, bioinformatics was a course I wanted to do my master's on, but I had no mentorship at that point in time because I love anything calculation, anything that has to do with logic, thinking, data. I love that. So I had no mentorship at that time. So that's why I didn't go into it. But going through this program, it has bring that back, and I also hope to, go further than that. Perhaps maybe after this program, look at towards taking up a degree program in bioinformatics to add to what I have, and see how we can start up a department in my college and university as a whole. That's my goal in all of this. So it's actually been an amazing time here, analyzing data, visualizing it. I was like, this is really good. So you guys have done pretty well. And the way you followed us one-on-one, you couldn't imagine Mr. Detail at 11 p.m., past 11 p.m., he was still taking me through certain processes at that time, late at night. So I like the way you followed up with the students. The mentorship was just there. It was very, very impactful. At least for me, it was very impactful, and I followed up very well. And I say congratulations and well done. And as we, advance to the other sessions, I believe they will get better and better. So well done, everybody, and well done to the team out there. Thank you.
Thank you very much, Nancy. That's a very good remark. We appreciate your kind words. And I need to say this now for us to take notes. This is organized by Eureka, the team that we have on ground, facilitators and mentors. And Eureka stands for Universal Research Knowledge Acquisition. And the concept behind Eureka partnering with Pine Biotech is to see how we can bring bioinformatics to our own language, to our level, and be better off when it comes to research. And we hope to keep this research team. Already, we need to let you know, it's a family already. And we want you to still be on ground to see other laurels that will come from this. There are better goodies that beyond the classes that you're going to see that we're looking forward to. So I would want to take one more response, then we're good to go. Do you have any person?
Good evening.
Good evening, Dami. Yes, you have the floor, please, briefly.
Yes, I want to say a big thank you to the organizers of this program. See, I started the cohort session like a week after the session had started, and I didn't even feel it that I just started. Like, I started late. I didn't feel it. You guys were always there, and it's amazing. The courses were actually down to earth, like we could understand it. I could understand it. So it's been an amazing journey so far. So thank you very much. The mentorship was just perfect. Thank you very much.
Wow, I'm blushing here. Thank you very much, Dami. That's a good one to hear from you guys. It's very, very encouraging, because the concept behind us coming together was to ensure, particularly Nigerians, start up a research community in Nigeria, and let's see how far we can push research. We want the globe to know that we have researchers that can do better from scratch. And that is the concept behind the Eureka team, to ensure that we have a very good outing. Distinguished ladies and gentlemen, at this junction, I want to call one of our very own mentor and our committee manager that has been relating with you very much well. He's going to take the 6th session, and by the end of this session, he's going to give you the gist behind the next item after this, because we have loads of activities that we're going to do together when it comes to research, because we want our name to be written among the first 10 in the world. I'm not talking about the first 100, the first 10. And to do that for us, today's session will be handled by Dr. Adetayo. Dr. Adetayo, you have the floor now.
Thank you, Mr. Oni. Thank you, everybody, for the kind words. I hope we'll be able to continue being a source of inspiration and being mentors to everyone in the community. The good thing is in Eureka, our motto is learn one, do one, then teach one. So that way, we are all learning as we go. We also keep learning, and the more we get to teach and train these participants in this community, the more we get to learn, and the more we get to go together. So straightforward, let's without further ado, let's move into today's program. So we'll start by having a review. What have we done so far in this cohort? Well, we started with basic of DNA and the central dogma of biology. We learned about different NGS techniques. Subsequently, we learned about the types of NGS sequencing. We talked a bit about sequencing widths based on length. We also talked a bit about rearrangements in chromosomal levels. We talked about types of mutations. We mentioned the missense, the nonsense, and once in a while, we also mentioned the frameshift mutations. Then from there, talk about what codons are and how a triplet codon actually gives rise to an amino acid. Then we talked a bit about the significance of DNA mutations or DNA variations in conditions like cancers. We mentioned acquired mutations, germline mutations, and somatic mutations. This we also went ahead to demonstrate in a genomic mutational analysis pipeline where we use Strecker after usage of bow tie to be able to call our variant. I think last week we went through the practical hands-on on how to view mutations on IGV, and I think we demonstrated that. Then before this, we've had a load of courses on our educational platforms where we have gone, each and every one of us, and attempted some of them. We've gone through introduction to bioinformatics, introduction to genomics, genomics 1. As our last week, our goal was to complete transcriptomics 1 and explore working with the expression table. We also observed that some people have not fully completed their profile. So we enjoined them last week to complete their profile. This is a community. We want to know our names, we want to be able to relate with ourselves. So knowing our faces, knowing our names, knowing our faculty, department, where we work, what we do, would enforce camaraderie in the community. That's why we always insist or try and appeal with our police. We should all complete our profile. So that's basically most of the things we've learned, basically on genomics. Today, we are going to talk about transcriptomics, which is like the next step. But before that, as usual, let's go through some questions to see how far we've come. So question number 1 comes, which NGS technique has the highest coverage? A, targeted, B, whole genome, C, whole exome. A, Boli, targeted, Banker A, Sawa A. Well, we are all correct. Targeted has the highest coverage because of the way it is sequenced. So our next question is, which type of mutations insert stop codon in the sequence? Which type of mutations insert stop codons in the sequence? Thank you, Abdulaziz. Thank you, Sarah. Thank you, Dr. Boli. Thank you, Promise. And the unanimous response is nonsense codon. I think we discussed this last week why nonsense codon is called the way it is. The next question is, frame shift mutations are always results of mutation only at single nucleotide position in the sequence. Is it true or false? Thank you, Dr. Boli. Thank you, Abdulaziz. Thank you, Levis. Thank you, Banker. Our unanimous answer to is B. Frame shift mutations may occur at more than one single nucleotide position, and that is right. And I think one more. Which of the following parameters provide us information whether mutation is significant or not? Let me repeat that question. Which of the following parameters provide information whether a mutation is significant or not? A, the position of mutated nucleotide at reference genome. B, mutated nucleotide base. C, somatic score. D, none of the above. Which of these following parameters provides information? Sarah said A. No responses so far. Dr. Boli said D. Elvis said D. Abdulaziz picked C. Dr. Nancy A. Ade D. The answer is C. It's the somatics core that provides us information about the significance of a mutation. I hope you are clear on that. It's the somatics core that will provide us information about whether a mutation is significant or not. A nucleotide can be mutated, and yet there will be no significant changes in the genome, or no significant changes in the susceptibility or in the condition. But the higher the somatics core, the higher the significance of that mutation in any heterogenesis or mutagenesis. Do we have any questions as regards that, or any queries? Okay, then if not, then straight to today's business. We're talking about the introduction to transcriptomics, gene expression data analysis. The first question researcher you should ask is, why transcriptomics? Well, as a biologist and as a researcher, the questions to ask are, what genes are present in the genome? We have been able to answer this by all the analysis we did the following weeks on genetic data. But there are more questions for a researcher or for a biologist. And they're like, what proteins are produced when these genes are expressed? What is going on in a particular cell within an organism at a particular time? How does a cell perform gene expression? And how does gene expression, how does it get altered in a cancer cell? All these questions leads us to transcriptomics, which means gene expression. It is a known fact that all individual cells in our body possess the same DNA, the same genomic data, all in our body. Dr. Nancy, can you hear me now? Am I more audible now?
Yeah, better.
Oh, okay. So if all the cells in our individual body possess the same genomic data, how come in the hair, the cells produce hair? How come in the blood vessels, what they produce is blood? How come on the feet of our skin, what they produce are the epithelium and skin? When all of them possess the same genomic data, it means that they all express genes differently. The genes that are expressed in individual tissue are not as a result of the genes that are expressed by that component. The genes expressed in the head and neck region is different from the genes expressed in the abdominal cavity. The same thing with plants and animals. The genes expressed by different locations differ. And this is the reason for transcriptomics. It means all of them have the same capacity to produce the same thing. But for one reason or the other, either due interactions with the environment, either through interactions with proteins, either through interactions with other transition factors, some genes are not being expressed. So a total of the gene expressed at a point in time, the knowledge of this is known as transcriptomics. And for a gene to be expressed, it must produce mRNA. And it is mRNA that is then translated into proteins. So it is then straightforward to say that transcriptomics is actually the study, the quantification of the mRNA production or expression in individual cells or tissue. By now, if you have gone through transcriptomics course, you should be familiar with the process of RNA transcription. Today, we'll just focus on how we can process raw RNA-seq to produce a structured data in the form of a gene expression table, and then apply different statistical analysis to derive biological significance of data. But first, let's have an overview of transcription process steps that takes place within a cell. First, it starts with RNA polymerase, adding complementary nucleotides to a DNA template strand. 2, various proteins bind to the polynation site near the three end of the pre-mRNA molecule. And next, a cap and a tail are added to the respective 5 and 3 ends. 4, non-coding introns are removed and the exons are joined together. By now, we know the difference between exons, which are the protein coding regions of the gene, and introns, which span exons. By the time DNA is being transcribed to mRNA, the introns are being spliced, leaving only the exons. And finally, the 5 cap is recognized by a nuclear pore complex, signaling the export of mRNA from the nucleus to the cytosol. Next, how can we quantify and detect mRNA? There are 3 major methods that can be done. Can anyone tell me the methods for RNA quantification? You can please post the answers in the chat. What are the 3 main methods? Yes, Abdulaziz? PCR, or you mean RT-PCR, which is reverse transcription PCR. Yes, RT-PCR. Thank you, Abdulaziz. That's one of the 3 ways. Any other answer? Microarray analysis. Thank you, Abisogo. That is also the second way. The microarray is similar to northern blot and is hybridization based. Spectrometry, not really. Then, Dr. Latunji, NGS, next generation sequencing, or RNA sequencing, or is the same thing. Farida, thank you very much. So we're saying that 3 major ways. Of all the other ways, reverse transcription PCR and microarray, there are ways that are used for known genes or for known mRNA transcripts. You must have identified the mRNA sequence before you can make use of either microarray or RT-PCR. NGS, transcriptome sequencing, is a major advance in the study of gene expression because it allows a snapshot of the whole transcriptome rather than a predetermined subset of genes. As I said earlier, for us to make use of microarray and RT-PCR, the subset of genes must have been predetermined. However, NGS or NGS transcriptome sequencing provides a comprehensive view of the cellular transcriptional profile at a given biological moment and greatly enhance the power of RNA discovery methods. To study this data, we will learn about processing pipelines that will turn raw reads into structured data ready for analysis. The NGS transcriptome sequencing is also very similar to NGS that we have been discussing all this while. The only difference is the starting point. For genomic data, the starting point is the DNA, while for transcriptome sequencing, the starting point is the RNA. That implies the first thing we need to do is to extract RNA from cells. And these RNAs are then converted to cDNA, meaning the RNA is reverse transcribed to DNA and is known as complementary DNA or cDNA. Then this cDNA is then fragmented, shared, and placed in a flow cell. Inside the flow cell, the DNA fragments are amplified using bridge PCR amplification, just like NGS genomic data sequencing, and the flow cell is inserted into the sequencer. The sequence machine uses image analysis to capture each letter in the flow cell fragment by analyzing visual patterns and converting them to a sequence of letters, either T, C, G, or A. Eventually, it will produce reads, typically 30 to 300 base pairs. And just like genomic data, sequencing data is stored in these several file formats. For example, FASTQ, which is not only text-based like FASTA, but will also provide us the thread quality scores of each read. FASTA is text-based format for representing DNA and protein sequences. And typically, the GTF, also known as, which is the genome transfer format, is what Mr. Adekunle spoke about during his own session. And we said that it contains genome annotation. For example, it will contain the start and end of each genomic element, each exon, each isoform, and each gene. And of course, the SAMBAM files, which are the sequence alignment mapping files, or the binary alignment map, this represents the alignment of each read to the reference genome. They usually start with the header section, followed by alignment information, and tab separated lines for each read. Now, to process next generation sequence data, we must map reads to the reference genome. To do so, we have to know where the exons on the genomes are. And this information is contained in the GTF file. Then we need to take the sequences from the FASTA file, where we have short reads, to align to the genome. The reference genome gives us information on where each gene is located and what exons are on what gene. Then as different isoforms are created from this single reference genome, different reads are mapped onto specific exons. And because they are mapped in different quantities, FASTA files that give the different sequences of the reads. So after running a pipeline of these genes and isoforms gotten from an RNA sequencing data would look like this. This is the old sequence. You can see these are exons that code for gene A. These are exons that code for gene B, while these are exons that code for gene C. It means the base pairs in between this are introns and not coding proteins. So with this gene A, B, and C, we can have different transcripts. You can see transcript, a gene can be formed from just transcript A, an mRNA can be formed from just transcript A and transcript C, while another can be formed from transcript B and transcript A. So all this so all these are then quantified and added, and the result is in the form of a tabular format. So let's quickly run through the overview in the pipeline. The first thing is preprocessing, just like we were talking about when we were doing our genomic mutation. One, the adapters are removed, and nucleotides with poor quality bases or low thread scores are trimmed. This is made use of with the use of chemomatic. Then the PCR duplicates are removed. This is made use of with PCR cleaner. Then the next step is mapping, on which the fragments or the reads are mapped on a set of known transcripts, mapped on the genome and identification of novel transcripts, or at times there could be combined strategy of both. Then after this, the pipeline quantifies the expression levels of each transcript in the sequence. And it does this by normalization of the total count in this format. When we get there, we'll talk about it. So that a typical pipeline, after start button, you do preprocessing with chemomatic and PCR clean, then GTF processing with cuff merge. Then they are mapped to the reference genome by the use of any of these three. We have Botai2, which by now we should be used to. We have TopArt and ISAT. Then after mapping to the genome, then the expression levels can be quantified using any of these apps, any of this software. You have the Sailfish, the ARI SEM, and the HTC. Then the isoforms are constructed by the use of cufflinks. And if you need detection of exons, this is done by the use of Augustus. So once again, the step one, preprocessing, data simulation and error correction. The step two is mapping on the reference genome. The step three involves calculating the abundance of reads aligned to the reference genome. While step four is biological interpretation based on quantification of the transcript in this reference genome. All available software are on so let's talk about each of them one by one. Reprocessing. It cleans data, reduces technical artifacts, and increases the likelihood that reads will align in the right location. And to do this, we have the Chemomatic and the PCR clean. Next is mapping. Mapping identifies the best place for a read alignment. It identifies splice junctions and visualizes mapped reads. To map, we have this software. We have ISA2, TOPA2, both tie, and their job is to align RNA sequences, map read across splice junctions, and align sequences read to reference genome. ISA2, both tie, and TOPA2 are mapping algorithms. They are not the only mapping algorithm. There are several mapping methods that are available, and they can either map on the full genome or manually detect alternative splicing. We can then use a known set of transcripts from the available GTF file. Botai2 is a fast algorithm that is based on a seed or KEMA approach. Well, what we mean by this is there will be substrings from the read, and their reverse complements are extracted and aligned to the reference genome in an orthogonal fashion. Then their positions are recorded and they are extended to full alignment using what we call a SIMD, accelerated dynamic programming. Another mapping software is TopArt. The other one is Bowtie. And any of the three can be used to actually map to the reference genome. Then the next stage is calculating the abundance of each reads that has been aligned to the reference genome. And once the reads are aligned, the quantification method counts the number of reads aligned to each transcript. It converts counts into relative expression levels. And this conversion can be done by using Sailfish, RSCM, and HTC. So that after the processing and mapping steps, expression values for genes and isoforms are generated and displayed in an expression table. The counts of expression are usually in read count, but some, or for most of them, they also express in terms known as FPKM, which means fragments per kilobase of transcript per million mapped reads. We'll get to that in a minute. But note that half of the time, what we have is not actually the read count. At times it is FPKM or TPM. And what do we mean by these terms? Now for pair end reads, the FPKM is calculated by number of reads mapped on the genome, divided by total number of mapped reads in millions and gene length in kilobase. Another term that can be used is transcripts per million, which is the number of reads per kilobase per millions of total sequencing reads. So this is an example of when we use a single-ended sequencing read, we call it RPKM. But when we use pair end, the term uses FPKM. So for RPKM, you use mapped reads multiplied by 1,000 bases times 10 to the power 6, all divided by length of transcript multiplied by total number of mapped reads. For in example 1, let's assume we have 2,500 kilobase transcript with 900 alignments. The RPKM will be 900 into 100.10 to the power 6, which will give a count of 45. So this is a visual representation of the RNA-seq pipeline in Turbo server. We'll go and do that in a minute. So from start, this is preprocessing, this is mapping to the transcript, and how this SCM table quantifies the reads, and we end. In total, the end result of this pipeline is an expression table, which looks like our typical Excel table. And on it, for the rows, you would have the genes, while for the columns, it will be sample or the variables. Depending on how many samples we have, the samples will become the variables, while the genes will become the rows. That implies at each sample, the quantity of genes quantified.
Hello, Dr. Adetayo.
Will be expressed
Can you
in
hear me?
numbers.
Dr. Adetayo, okay. Your line is breaking.
Oh, can you hear me now?
Yes, we can hear you now. You can go on.
Okay. I said before we go over to the pipeline, are there any questions as regards the steps involved in RNA-seq? You can put them in the chat section. Any questions?
Yes, I have a question. Sorry.
Okay, no problem.
Please, can you go back to the formula, the RFPK?
Okay, a second.
Yes, thank you. So the number of reads mapped on the gene, what's the difference between that and the total number of mapped reads in millions per kilobase?
The number of reads
mapped on the gene.
For each gene, depending on the coverage of that gene, there's a number of reads that is mapped to a gene, right? Okay. And there is total number of all the mapped reads, right? We are saying each gene has transcripts. A gene can have like 10 or 20 transcripts, right? That implies the longer the gene, the more reads are mapped to that gene, right? Let's do it this way. It's called balls in a marble or marbles in a bag. If you have a lot of colored balls in a bag, every time you dip your hand, and let's assume you have 10 red, 2 blue, right? So every time you dip your hand into a bag, there is a high likelihood that you bring out the blue color, right? Or that you bring out a red color because you have more red than blue. Do you understand? Hello, Dami.
Yes, I understand. Thank you.
Oh, okay. Any other question? Any other question? If you are not, then let's quickly go to the pipeline. I want please, can you see my screen?
Yes, we can see your screen.
Okay. So there are two ways. We can either use one of these demo pipelines here, or we can follow Transcriptomics 1 course where we can import our own file. I'll try and do both. I'll try to do both fast so we can go ahead. For the demo pipeline, I would like to make use of this RNA-seq, which is a project you should be familiar with by now. It's about breast cancer, trying to look for variations or differences between the triple negative breast cancer and the ER positive breast cancer. So we'll load this into the pipeline. Once we load it, then we'll go straight to start. From here, we can see our input files. You can see the input files that includes the links and the reference genome. So the next thing is highlighting for us is Geomatic to trim the poor quality reads. We'll click that. The next thing is PCR clean to clean the PCR duplicate. Then from here, we can see that the software that have been highlighted includes the bow tie, which maps to the genome, the ISA2 and the bow tie 2T. But because we are mapping to transcripts and not to genome, we'll pick the bow tie 2T. And from there, the next thing being highlighted is the RESM expression table. In case we want to do differential expression between the two cancerous conditions, we can use this, right? But for now, we just want to see our expression table. So we just click end. And that will be the end of the pipeline. Because it's a demo set, you can see the results, right? These are the results of our expression table. For the expression genes, this is 1. These are only with counts that have not been normalized. But these are with counts that have been normalized for FPKM, right? But you can see there are 2 FPKM. One is FPKM.txt and one is FPKM not filtered. What it means is, at times during expression tables, during quantification tables, this table only consists of the counts. For some samples, the counts are zeros all through. And we know having a lot of zeros will add only outliers and might affect our downstream data analysis. So this has been done in such a way that all the zeros, all the samples that present zeros are filtered away, and we only have samples that have at least either 5 or 6 reads. But depending on what exactly your research question is, you can either make use of this or this. This will be the result of our output. Is there anyone? Are we all here? I hope I've not been too fast in the processing. Okay. Let me go over with the one described in Transformers 1. If you have your own file or you have your own data, you want to process it, you don't need to pick any of these drop-down buttons. You can click this directly, right? And just like our genomic data, because we want annotation, we have to pick genome GTF file, which is already picked. Our end reads is pair end reads. We are making use of FASTQ format. And as usual, our genome is human mouse concatenated. Then you can do file upload, right? There is a bulk upload link in Transcriptomics 1. So you can click this. And if you click this, you search for where you put it on your system. So here is mine. Right? So I've added it. And now, next thing is to keep group selection. At this point, I'm actually not looking for differences in the expression of both. I just want to see the quantification. So I don't need to worry about group selection. I'll just cancel and say no groups and continue, right? Are we together to this point? Are we together to this point? Are they? Okay. So from here, we click start. Click OK. Let's assume we don't want to do all this because we have already done it in the past processing. From here, after doing this, which we won't bother ourselves with now, we can just go straight to BOTI2, right? BOTI2 will map to the reference genome and map to the transcript. Then from there, the next thing is quantification with RCM table. You can see our option here. If you want it FPKM, you can pick. If you want it in just count, if you want it in TPM. But for now, we'll choose FPKM and save. The next is what? We hand the pipeline. Then we'll save. You can give it any name. Let me use. Then we'll click one pipeline. And as usual, this pipeline will take a while. So it's not something I would expect to be done in few minutes. But in preparation for today's session, you can see that I already ran one and I titled it for today's session. So I'll just go ahead and open this. It was the exact same pipeline, and let's see the results, right? So the results, these are the output files, pipeline output files and downloadable files. I'll click on this. And I said earlier, we have FPKM not filtered. We have the genes text. We have FPKM. We have all these options. Let me go ahead and click this. So this will be downloaded. So let me go to my downloads. This. I'm going to open in Excel format. Okay. Let me do this in a bit. So are we all here? You can see that this number 2, this is a gene ID, right? And this gene ID in this sample is 0, 0. In this sample is 12.95. Let me do this. All these are ER samples. And all these are triple negative. Let me color them differently. Let me color them red. No. No. Okay, it's better than that. So from now, what it means is for this gene, these are the expressions in the ER positive samples. While these are the expression profile in triple negative samples. So all the genes, if you go this way, it's almost, this is a lot of genes. I'm sure we have close to 6,000 or 9,000 genes or thereby. But what we can see from the running of this pipeline is a tabular format where each gene is expressed with numbers in each of these samples. Are we together? Are there any questions? So that what it means is at the end of the transcripts in each of this, meaning this gene 2834, this is his count in this sample. This is the count in this sample. And these are the counts in these individual samples. So once we have this table, we can then begin our downstream data analysis, which will focus more or will deal with better when in the next cohort. So let's go back to our slides. So the end result is this gene expression table with genes written in their assemble ID and the columns in samples. So at this point, you have to ask, are there questions or are we clear to this point? Elvis says, how do we know which of the genes are AL or ER2 genes? All I see is Elvis, this is very easy. The genes are written in their ensemble ID, right? If you want to know what each gene represents, you can just take one of these genes, go to ensemble. You can even go on Google, type this ensemble ID, and the name of the genes will be displayed, right? At this point, when we are done with the analysis, when we finally get to a point where we know the significant genes, where we know where we can identify or characterize the gene we want, we can go ahead and do what we call annotation, gene annotation. But at this point, doing that's some analysis we want to do, we really are not so concerned about the name of the gene at this point, about the annotation of those genes at this point. The end result of this is we can do gene annotation, we can do systemic network to be able to not only know the gene, but also be able to identify the pathways in carcinogenesis or mutagenesis of the conditions we are interested in. But if you want to go ahead and just know what does this mean, these are ensemble ID. You can copy it, put it on in ensemble or Google, and the name of the gene will be characterized. Is that clear, Elvis? Okay, Oboli said, okay. Dr. Oboli, what we said is, after running this pipeline, you have a couple of options. They can give you just read counts, meaning just the count of transcripts of each gene. But this will not be normalized. This will not give you the overview of what exactly is happening. With read count, it stands to reason that genes that are longer in length will have more widths, and genes that are shorter in length will have shorter widths. Then what it also means is if two different people are doing two different types of NGS, and one NGS has an higher coverage than the other NGS, then it stands to reason that there will be more count for the sequencing that has higher coverage than the low count. So read count needs to be normalized. And the ways and manner in which they've been normalized is this quantification format, which is FPKM, if it's pair-end read, or RPKM, or this transcript per million. And this is the formula. The number of reads mapped on one gene is taken and divided by the total number of mapped reads in millions, multiplied by the gene length in kilobases, meaning number of reads on a gene, the count, divided by the total number of mapped reads in millions, then multiply it by the length of that gene in kilobases. So this will give the exact count of that gene, and this you can use across genes or across samples, right? So if it's a single end, you call it RPKM, and this will be the formula. So this way, you can compare different genes across different experiments, or compare counts across different genes, because you have normalized based on gene length, you have normalized based on total number of mapped reads, and you have also put into cognizance the coverage of that gene, or the coverage of the NGS sequence. Dr. Oboli, is that clear? Okay. Okay. So if there are no further questions, so then the next question to then ask is, all we are just seeing is numbers. How do we visualize this? How do we visualize the quantity? And what we usually use for this is box plot, right? And I'm sure most of us that have done R, we've done data visualization both in R or in Python. At site visualization, we can get a lot of meanings from these reads. We can identify a lot of pattern. We can identify expression patterns in different conditions. We can maybe from 2 different maize plants, or from 2 different microorganisms, or from 2 different cancerous conditions. We can identify patterns. And learning observation of pattern, we can also identify markers for each condition. So this is just the preliminary. This is just a tip of the iceberg. There is much more in transcriptomics. But because at this point, this is a basic course, our plan is to introduce us gradually and progressively. But by the time we start the next course, we are going to continue at this point, and we are going to learn how to visualize an expression table. We are going to learn the biomedical data analytical methods that can tell us differences, that can tell us not only how to note differences between conditions and samples, but also give us an insight into biomarkers that can be used to make diagnoses for a sample. And from there, also tell us about clustering, about machine learning applications in biological data. So I sincerely hope you all join us in the next course. And from there, we'll start with transcriptomics. Of course, we also plan to talk about metagenomics, but these are many more. I will leave this to the program director to give us an hint about what to expect in the next course. So at this point, I would like to hand over to Mr. Sonny, who will then invite Mr. Adekunle. Mr. Sonny, over to you.
Thank you very much, Mr. Adekunle, Dr. Adetayo, for the very, very insightful discussion you have given us so far. And I'm sharing my slide now, right immediately because of time constraint. And I want us to be able to manage ourselves as much as possible. Hello, can you see my slide, please?
Yes, we can.
Yes, Dr. Adetayo had done a very good job taking us through that destination. And moving forward to the next level, which we need to discuss, is after this particular cohort, we are moving to the next level, which is the basic omics 2 training. And this will focus on process analysis of high throughput data sets with the logic behind pipelines and project-based learning for bioinformatics in research, agriculture, and precision medicine. Now, for us to go into this second cohort, looking at the second training, part of this particular training we are having so far, we are going to have 3 sessions that will come on ahead before the main session. This is to prepare us for the training 2. And what we are looking forward to is that we're going to have 3 webinars. The first webinar will be holding on like February 24th, 2021. And second webinar will be sometime in March 9. And finally, we're going to have another webinar on 16 March 2021. Now, I want us to take note that the essence of this webinar is to serve as a precursor to the main aspect, so that those that are yet to be part of this, those that are not part of this particular cohort, can blend in and go with us. While those of us that are already in this cohort, it will be an easy way for us to dot our I's, cross our T's, and move to the next level, which we are looking forward to, that the basic training will be starting 23rd of March 2021. Now, for the basic OmicsLogic 2 training, what we have on our screen is a schedule. And the schedule, as you can see, we have what we know for session 1, NG's data and genomic sequence analysis, which will be our first session. We're also going to take